Move on now to our next speaker, who is Dr. Bali Pulendran, the professor and director of ITI. Bali completed his undergraduate degree at Cambridge University and PhD from the Walter and Eliza Hall Institute in Melbourne under the supervision of Sir Gustav Nossel. Bali has been a pioneer in systems approaches to characterizing immune responses during infection and vaccination, and he's made several foundational discoveries related to dendritic cell biology, characterizing different functional subsets and activators of maturation. And he's going to tell us today about new work and universal approaches to immunity. Thank you, Bali.
Thank you very much, Taya. And again, it's great to see the room filling up here. And so I would like to talk with you about some unpublished work. We have this in revision in a journal. And the idea is that you could program the immune system in such a way that it provides essentially pathogen agnostic immunity in mucosal sites for a few weeks to a few months. A major focus of the work in our lab is to use vaccines as probes on the human immune system to essentially decode the inner workings of human immunology, but using vaccines as probes. And we and a number of other labs have really used this approach over the last few years to begin to understand the molecular networks that are induced by vaccination and eventually some new human biology that's come forth from this. But today, I'm not going to focus on that. Instead, what I'd like to talk with you about is the idea that the innate immune system has a form of immunological memory. And really, the genesis of this idea comes from work started by Mihai Netia and colleagues in Europe, the idea of trained immunity. So when I first came to Stanford back in 2017, we were very interested in this idea, and we wanted to see if this applies in the context of vaccines. So we did this experiment in humans where we immunized healthy adults with the H5N1 AS03 adjuvanted vaccine from GlaxoSmithKline, and then did this single-cell RNA sequencing and single-cell ATAC sequencing of blood samples derived from these individuals before they got the vaccine, and then 1 month or 2 months after they got the vaccine. So this was published in this paper back in 2021, but the essence of this result is shown here. In blood monocytes, particularly classical blood monocytes defined as CD14 high, CD16 negative, there were these remarkable epigenetic changes that seemed to persist for up to 2 months after the second H5N1 AS03 vaccine. It's a 2-shot vaccine. And these changes could be defined in this way. Chromatin loci targeted by the interferon regulatory factor 1, 2, 7, 8, and 9, as well as STAT1 and 2 were open. Loci targeted by the AP1 transcription factors were closed, like C4C June. This was of interest to us because we knew that these transcription factors are very important. The interferon transcription factors in an antiviral program, the AP1 transcription factors in a pro-inflammatory gene expression program. And so the question was, is there a functional correlate of these epigenetic changes? And what we did to analyze this was to isolate blood monocytes from people before they got the vaccine, but also 2 months after they got the second vaccine, 1 month or two, at day 42. And we infected these cells with blood-borne viruses, which we know have a tropism for myeloid cells, namely dengue and Zika, very different viruses from the H5N1 bird flu vaccine virus. And what we could see was that there was indeed a better viral control of these viruses in vitro, suggesting that these epigenetic changes may have imparted some degree of resistance, broad resistance, in a virus-agnostic manner to these viruses. Since that time, and I should say this work is led by a wonderful, who was a wonderful postdoc, Florian Wimmers, in collaboration with Nir Yosef and Ruchir Rastogi, and Florian is now an assistant professor at Tübingen. And so we're now working on an extension of the study where we look at a bunch of different vaccines, including the Pfizer mRNA vaccine, live attenuated yellow fever vaccine, and Shingrix vaccine, and asking whether these epigenetic changes we had seen with the H5N1 vaccine, are they more broadly applicable across a bunch of different vaccines? I'm going to show you a couple of slides from this, but basically, it is indeed in this classical blood monocyte population that we see the most profound alterations in the epigenome after vaccination. And so what we've done is to ask vaccine by vaccine, what are the changes in the epigenome, but also do a meta-analysis. If you now coalesce all 6 vaccine data sets into a single data set and ask what are the broad epigenetic changes that are happening within this classical monocyte population? Is there a certain pattern? Is there an organization to these epigenetic changes? And this can be summed up in this heat map. Here you can see in the bottom right are the loci targeted by AP1 transcription factors. And I should say, this is single cell ataxic data in classical monocytes. You can see this is red, meaning this loci is wide open, 21 days or 42 days or a few weeks after vaccination. Interestingly, however, if you go up here, you see loci targeted by IRF and STAT1 loci. They're all blue. In other words, there seems to be a reciprocal pattern in terms of how these epigenetic, what we call super motif clusters, are regulated. So when AP1 opens up, IRF closes. And conversely here, when the IRF loci open up, the AP1 loci close. This seems to be broadly applicable regardless of the vaccine that you look at. So there's a certain organization in the epigenome in terms of how they're regulated. And of course, we know the IRF motifs confer antiviral resistance. The AP1 motifs confer more of an inflammatory response. And so the obvious question is, do these really correlate with inflammation, the canonical inflammation pathway versus the antiviral pathway? Also of interest is if you look at this seemingly homogeneous population of classical monocytes, which is CD14 high, CD16 negative, if you do the single cell ataxic, you can see at least 5 different clusters that are epigenetically distinct. In blue, you can see the C0, the ISG positive cluster, which expresses the antiviral program. And in red, you can see these AP1 clusters. And so the question is this of any functional consequence? Well, the first remarkable finding was that when we looked at this across individuals, different healthy human, seemingly healthy human beings between the ages of 18 and 45, and each column represents an individual. And what we're looking at here is the relative abundance of each of these epigenetically distinct clusters within the classical monocytes. And you can see here on the far left is a person who has very little, if any, of the C0 cluster. But on the other hand, you look on the far right, this person has an abundance of these clusters. So the question is, this is at baseline. This is before anyone ever gets the vaccine. The question is, why is there such a wide variation in the relative abundance of these epigenetic clusters? What does this mean functionally? What does it mean when you get older, and so on and so forth? The other important question that we have to address as we talk about these epigenetic changes in innate cells that last for weeks, months sometimes, is that and these blood monocytes have a very short half-life. Heavy water labeling in humans suggests that it's 1 to 2 days. So how is it that you can have such long-lasting epigenetic changes in such short-lived cells? So one obvious hypothesis that's been put forward is that these epigenetic programs are imprinted at the level of the bone marrow precursors in the bone marrow by the myeloid precursors. And we know that these develop from the CMP or the common myeloid precursor, which can then differentiate into GMP, which is granulocyte monocyte precursor, or the MEPs. And so we wanted to address this question. And so what we did was to obtain human bone marrow or human blood from the same individuals before they get the vaccine, and then a month after they get the flu shot. And then we perform 3 different techniques on these samples. Number 1, single cell RNA sequencing, single cell ATAC sequencing, but also together with Ansu Satpathy in his lab, we perform these mitochondrial DNA sequencing. The purpose of this technique, and this was really pioneered by Aviv Regev and some others, and by analyzing the pattern of mutations in the mitochondrial genome, you can begin to infer parent-progeny relationships between cells. Of course, these cells do not express T cell receptors or B cell receptors, so we have to rely on this other technique. So the first question is, in this new set of experiments, can we confirm our earlier findings? And it's always very nice to be able to confirm one's own earlier published findings. And so thank goodness, here we are. So this is 28 days after we give the flu vaccine. We're looking at blood monocytes, and there you can see the IRF loci opening, the AP1 loci closing. So that's very good. We've confirmed it. Now, what about in these bone marrow precursor cells, the myeloid precursor cells from the same individuals? You can see that in the GMP, again, the same pattern happens as you can see in the CMP as well. Now, the question is, so this certainly correlates with what's happening in the blood, but is it really, can we define that mechanistic proof that it's the same cell, or can we define a clonal lineage that you can trace from the bone marrow to the blood? Now, this is something that we're actively working on now. Suffice to say that in the bone marrow, we can begin to identify different clones of CMPs and GMPs and begin to ask how many clones are there in this compartment? Is there evidence for clonal expansion? And if so, what is the basis of this clonal expansion? Just to give you a sense of this rich human biology that can come forth from this kind of multi-omic studies. Now, in the second half of the talk, I'd like to disabuse you of the notion that it is all about epigenetics. It is fancy to talk about epigenetics, but we feel as important as it is, the human immune system is far more complex. It's a hierarchy of different cell types that exist, all interacting with each other. And phenomena like immune memory, we believe, is really an emergent property that lies at the interaction of the adaptive and the innate immune cells. And that sounds very fuzzy and vague, but I'd like to give you two examples that speak to this issue. The first example comes from a study that we published a couple of years ago, where we looked at this BCG vaccine against TB. It's one of the oldest vaccines, and it's given to every child in the developing world close to the day of birth or on the day of birth. We in America don't have it. I've had it. I have a scar mark to prove it. The thing is that there's epidemiological data that people who get the BCG vaccine, populations vaccinated with BCG, seem not only to be protected from certain forms of TB, but also have a reduction in all-cause mortality against many other kinds of infection. And it's a series of studies that have shown this, and it's been a rather odd and peculiar finding. Nobody really understands the immunological basis of this, but based on the idea that there's trained immunity, it's been speculated that BCG might work through trained immunity. So Audrey Lee, who is a wonderful PhD student in our lab, she's now in Singapore, was interested to dissect this mechanistically. So she did some experiments in mice where she immunized C57 black 6 mice intravenously with BCG, and then 21 days, 42 days, various time points afterwards, together with Mehul Sutha at Emory, we infected these mice with the SARS-CoV-2 B.1.351 strain, which is the so-called South Africa strain, which has a very high affinity for the mouse ACE2 receptor. So it does cause a reasonable infection in mice. And the first thing to do is to measure weight loss. And you can see nothing much happens in the first 2 weeks, but look what happens at 21 days, 28 days, 42 days, the effect is still there, although it's waning. And perhaps even at 3 months, there's a significant effect, albeit a modest effect. More strikingly, if you look at the viral loads in these mice, you can see that in the lung, there's really a substantial reduction in viral load, 93-fold at 21 days, 435-fold at 28 days, 92-fold at 42 days. Nothing whatsoever happens in the nose. In other words, protection, whatever this mechanism is, seems to protect against virus replication in the lower respiratory tract, but it really doesn't prevent infection. Now, this is published, so I'm going to summarize this in a couple of slides before I get into the unpublished work. The question is, what is the mechanism of this protection? Why does BCG protect against such a different pathogen? And so we did a number of techniques to analyze this, and I'm just showing you one example of this, where we did spatial transcriptomics. This is a mouse lung, an unvaccinated, challenged mouse lung. We use spatial transcriptomics, but also a couple of different antibody markers. Pancytokeratin stains the epithelial cells. CD4, which you can't see very many of because this is an unimmunized, uninfected mouse, is in pink. Look what happens a month or a few weeks after BCG IV immunization. There's a massive infiltration of these CD4 T cells. Now you can gate on these pink regions of interest and ask what genes do those T cells express. The number one hit is interferon gamma. That's not a surprise because BCG is a very good vaccine for inducing CD4 Th1 cells. So that merely confirms what others have shown. Now, The question is, what impact do those CD4 T cells have on the rest of the immune system, namely the innate immune system and the epithelial cells in the lung? So what we did is a third group of mice where we immunized with BCG, wait for a few weeks, inject an anti-CD4 antibody to get rid of the CD4 T cells, and you can see the depletion is really quite effective because you can't see very much CD4 T cells. But now you can ask, how has the depletion of these CD4 T cells altered the gene expression pattern in these epithelial cells, which are the targets of SARS-CoV-2 infection? And you can see here in the unvaccinated mice, there's really nothing in terms of the ISGs, or interferon-stimulated genes. BCG immunized mice have lots of this, but look what happens when you deplete CD4 T cells. I'm abbreviating a lot of other pieces of evidence. We did single cell and so on and so forth, but just to get the point. Now you can do your quantification with your favorite gene of interest, and the results are really quite striking. So this led us to the idea that protection in a mucosal organ is dependent on not a single facet of the immune system, but rather a synergy or convergence of the adaptive and the innate system. Now, we knew that BCG immunization gives you this quick early burst of innate immunity. That's typical of any vaccine. But what was surprising was that we did see this delayed, more sustained phase of innate immunity. And I've shown you a little bit of data that suggests that this delayed second wave of innate immunity is because of these T cells, the Th1 cells, which produce interferon gamma. We blocked interferon gamma, and that seems to imprint this antiviral program in epithelial cells and dendritic cells and so on in the lung. So this was wonderful because at the time that we published this paper, identical results by the work of Maz Devangi and Kim Tran, his postdoc who's first author on this paper is here, sitting at the back. They were published virtually back-to-back in the same journal. And Alan Scher's group had exactly the same results. And it's one of these things where it's gratifying to see results converging by different groups as well. But nevertheless, so we were asked to write a little one-page synopsis or a perspective on this. And so we coined this term integrated organ immunity. And the question was, it raised the question, how could we apply the immunological lessons of BCG to design new vaccines that would effectively confer broad protection? Clearly, nobody's going to advocate giving people BCG, IV. That's not very translatable. So here we said that this concept of integrated organ immunity can explain how the innate and adaptive systems and non-hematic poietic cells can interact in tissues to generate enduring protective immunity against diverse pathogens in an antigen-agnostic way. Considering immune responses through this framework could enable the design of a new class of vaccines termed universal vaccines. All right, it's easy to write a perspective, much harder to actually provide evidence for it, right? So that's what I'd like to talk to you about. So basically, we've been working with a company in Seattle called AHI to design a mucosal vaccine that's given intranasally that essentially engages the same pathways that BCG has been using. And so, this mucosal vaccine is very simple. It consists of a liposome that contains two different TLR ligands, a synthetic TLR7/8 ligand called 3MO52, and a synthetic TLR4 ligand called GLA. Why these particular ligands? Because of prior experience that we and AHI and many others in the field have. Now, if that were the only component, you would say, well, that's a terrific innate stimulator. But what we've learned from BCG is that innate alone is not enough. You need this T cell response that could provide and sustain protection. So what better antigen to use in mice than the world's best antigen, which is ovalbumin, right? So what we did, and these are all preclinical studies in mice, and we're now doing a non-human primate study, but also planning a human challenge study based on these results. So these mice were immunized intranasally with this liposomes that have GLA and 3MO52 at 0, 7, 14, 21. Why so many immunizations? Well, BCG is a live bacteria that lasts inside your macrophages for weeks, months. And we wanted to come close to that. And so as a first approximation, proof of concept, we decided, let's go overboard and let's give 4. We've done this, we've dialed this back to 1, 2, or 3. 1 doesn't get it. 2 is more or less good. It can get you what 4 does, but just to be on the safe side, we've used 4. And then 21 days, 42 days, or 3 months later, we infected with SARS-CoV-2. There you go. That's a weight loss. That's significant protection. It's waning at 3 months. Now, At 21 days, there's up to a 700-fold reduction in the live virus load in the lung measured in Nehal Sutha's lab. So not a trivial reduction. You can see the nucleoprotein here in NP, and you can quantify that. You can see there's reduction. You can do the immunohistology, H&E section, massive infiltration upon infection in unvaccinated mice, but in the vaccinated mice, the alveolar structure is very well preserved. We've also done this with the seasonal PR8 influenza strain. You can see 21 days, 42 days, 3 months, and even 7 months after the last immunization. And you can see measured by weight loss or survival, substantial protection that seems to last up to 7 months, although the effect is waning by 7 months or so. Now, with Ralph Barrick, we extended this to a bunch of other viruses. So we challenged with the mouse-adapted SARS-CoV-1 strain. There's protection against the SHC014 strain. There seems to be protection against that. So a bunch of respiratory viruses, which are really from different families of viruses. What about respiratory bacteria? So we also did this intranasal challenge with Staph aureus or Streptococcus pneumoniae at 21 days, 42 days, or 3 months later. And you can see with the Staph aureus, by 21 days, about a 14-fold drop. Same thing at 42 days, and by 3 months, it's waning. It's about a 6-fold drop. With strep pneumonia, there's a 20-fold drop, and then even at 3 months, there's a pretty significant 66-fold drop. All right, so what other kinds of respiratory threats can you imagine? We've looked at bacteria, viruses, and so on, and we did allergies. So we can do this with house dust mite allergen. So this is 3 months later after you give the vaccine, and you can see the unvaccinated mice induce a prototypic Th2 response with the eosinophils IL-4, IL-5, IL-13. But look what happens to the vaccinated mice. And the immunopathology is also improved, as is the IgE response. So how the heck is this happening, right? So what we looked at was innate responses in the lung by flow, and you can see AM, alveolar macrophages, which are CD86 expression on these cells is very low at day 0, increased at day 21, and at day 90, it persists. And you also see a similar effect in DCs, but albeit to a less pronounced degree. What about T cells against OVA? So we're lucky we used OVA because we have class 1 and class 2 tetramers. And you can see magnificent class 1 tetramer positive T cells that seems to persist substantially above baseline, even at day 90. You see these class 2 positive tetramers. And if you take the lung mononuclear cells and do an ex vivo stimulation with synfecal or class 2 OVA peptides, you can see stimulation of interferon gamma by these cells. Now, what kind of T cells are these? Are these effector T cells? Are these tissue-resident T cells? So we did CD45 IV labeling, and it turns out that vast majority of these cells are tissue-resident memory T cells. Now, the question you might say is, well, do you really need T cells? So what we did was 2 experiments. Firstly, we immunized these mice without the antigen, so there should be no T cell response, or with the antigen. And then we challenged with COVID at 21 days, 42 days, or 3 months. You can see at 21 days, it really doesn't matter if you exclude the antigen, you can still get pretty good protection. That's in pink. 42 days, that protection is waning, and by 3 months, unless you have the antigen, protection has gone completely. So what we think here is happening is that the first 2 to 3 weeks is what we call the prototypic trained immunity, which is kind of the epigenetic part of it. The time beyond that, up to 3 months and sometimes 7 months, is the integrated organ immunity that feeds back on the innate system to keep it alive. And we did this experiment the other way. In the interest of time, I'm skipping through this, where we deplete the CD4 T cells and show whether it's with Staph aureus infection or seasonal influenza infection or COVID infection, you absolutely must have T cells in the lung. And the way this seems to work is that those tissue-resident T cells are providing a signal that maintains these alveolar macrophages in an activated state. So CD86 disappears if you deplete the tissue-resident T cells. Now, the obvious question you're asking is, what kind of signal are these T cells providing? In BCG, it was interferon gamma, because those T cells are effector T cells, because they are seeing the BCG antigens for a long time. BCG lives inside alveolar macrophages. This vaccine is not BCG. It doesn't live inside alveolar macrophages for months. So we spent 1 year trying to identify this. We neutralized gamma, TNF alpha, CD40 ligand, 0 impact. But when we looked at the spatial and RNA sequencing data, we discovered rank ligand was produced by these T cells in abundance. So let's do the experiment. We immunize, but we neutralize the activity of rank ligand with this antibody, and you lose the protective effect with COVID, with flu, and bacterial infections. And the last experiment was, can you nail down that it is the alveolar macrophages? What we did, two different styles of experiments. One is to deplete the alveolar macrophages in vivo with clodronate liposomes, and then infect with PR8. You lose that protection. Same thing with Staph aureus. You see, if the alveolar macrophages are intact, you have great protection. But with clodronate liposomes, you lose those cells, you lose protection. And how are these alveolar macrophages conditioned? Interest of time, I'm not telling you, showing you too much of the data, but they have an antiviral program. They express ISGs, they express CD86, but they're also able to uptake phagocytose bacteria. So we can do this with labeled Staph aureus, or dead cells, whether it's green apoptotic cells or necrotic neutrophils, or virally infected epithelial cells. So, and in each case, these cells have this remarkable property. And I promise you, the last piece here is that I've told you that there's a profound 690 plus fold reduction in virus load, 15 to 66 fold reduction in bacteria. By no stretch of imagination is that sterilizing immunity. It offers a barrier to the pathogen, but many pathogen entities slip through that barrier. So the question is, what happens once the pathogen comes in to the pathogen-specific immune response? So we looked at this with SARS-CoV-2, but also with flu. We vaccinated mice or not, and we infected them with SARS-CoV-2, and 3 days later, 3 days is a relatively short time after infection, we did Codex, so this fancy 25-color technology that we have here, we can do imaging. And you can see in naive mice that receive SARS-CoV-2 infection, you can see the B cells and the T cells and the helper T cells, et cetera, et cetera. You can see viral antigen. Look what happens 3 days after vaccination with ovalbumin and liposomes. This is actually, so we're leaving a month or 2 months after the last vaccine, and 3 days after SARS-CoV-2 infection. These ectopic secondary lymphoid structures that are forming so quickly with beautiful organization. You can see the B cell zone here, the green helper T cell zone with the odd CD8 T cell sprinkled in there. How is that so? Is this virus specific? Is the immune system primed to launch so rapidly to this virus? So we did the tetramer analysis, this time looking at class 1 tetramer against influenza. You see 0, 6 hours, 1 day, 3 days, 7 days after infection. There's this enhanced CD8 T cell response, and there's also an increased antibody response. So it appears that this vaccine has not only, it's a double whammy. It's not only prevented, or I should say, reduced the pathogen burden coming in, not sterilizing immunity, but when the few pathogens do slip through this initial bulwark, it sets up the immune system to launch a super rapid, adaptive, antigen-specific response to this pathogen. And so this summarizes what I've told you. I'm not going to read this. Basically, this effect seems to last anything from 3 months to 7 months or so. We think it's a feedback of these memory T cells in the lung on the alveolar macrophages working through the rank ligand. We think that the alveolar macrophages are central. We can deplete them. We can put them back from a previously immunized mouse into a naive mouse and regain protection. And there's this remarkable feature of forming these very specific secondary lymphoid structures and a specific adaptive immune response after pathogen entry. So we think that this result may hint at potentially a new class of what we call universal vaccines that are capable of defending against a wide variety of respiratory pathogens and allergens. So we're planning studies in ferrets. There's a non-human primate study that's underway. We're also planning a study in humans where we can give GMP material to humans and then a month or 3 months later, infect these people in a controlled human infection model challenge. We're planning to do this at Imperial College in London. So I'd like to thank all the people who did the work. Haibo Zhang, who's sitting over there, is a brilliant postdoc who's just really led the way and just been super. She's only been in the lab for 2 years, but all of this she did effectively single-handedly. So, and then the early work on epigenetics was done by another wonderful postdoc, Florian Wimmers, and there were many others in the lab who helped us. So thank you very much.
Thank you, Bali. That was wonderful.
Yeah.
Hi, Bali. That was great. That was really wonderful. Yeah, I'm just wondering, do the T cells, the TRM that are generated from the vaccine, they are responding to the antigen? And also in that last part, are you generating BALT then from the vaccine? So in fact, I mean, have you measured antibody and such?
Yes,
Is
yes.
that, yeah.
We have measured antibody. I didn't show you, it's in the paper, but we do see a very rapid induction of IgG and IgM antibody within 3 days, 7 days or so, much more rapid than in a mouse that hadn't seen this mucosal vaccine. And your other question was, are these TRMs that were over specific, are they responding to the new virus, right? We haven't looked at that. We should certainly see if somehow they're able to become activated through inflammation or something and they're responding. Yeah. Ed had a question, and then yeah, sorry. Yeah, Fadi. Yeah.
Really fascinating observations. A simple question: if you leave out the ova, how much do you compromise the whole impact? In other words, is it possible that the reason we use adjuvants is actually justified here, but maybe the adjuvant should be given sometime before the adjuvant plus antigen is given?
Yeah. Yeah, it's a great question. We did do some experiments where we did leave out the ova. We can see the adjuvant alone provides protection for about 21 days, but beyond that, you lose the protective effect. Exactly. And I think Fadi, sorry. Yeah.
Do you think it provides any protection against lung cancer?
It's a terrific question. I mean, this whole idea of integrated organ immunity, getting the T cells, the innate, and the antibody, as you'll hear Taya talk about through the FC, we have to look at this in the context of cancer, infectious diseases, different systems,
So, Bali, fascinating
Yeah.
story. So how come the BCG trial in COVID failed?
Yeah, it's a
So
great question.
do you think you're doing something better than BCG?
Yeah. The easy answer, and if I want to be flippant, I can say mice lie and humans don't, right? And that's probably a huge element of truth in that. The other, I would say, is that the BCG trials use ID, intradermal. That's the standard route of BCG. We tried intramuscular and sub-Q. You can't do ID in mice very well, but that doesn't seem to work. It's the BCG IV that seems to have the best effect. The third thing I'd like to say is that you notice that there was no prevention of infection. The virus load in the nose was similar in unimmunized versus BCG. It was only in the lung. So the readout in the classic paper in New England Journal of Medicine that showed failure of BCG was really proportion of people infected. There were so few severe cases to get a real statistics on disease severity. So for all these reasons, I mean, that study was done beautifully well, but I think it's open to interpretation. So.
Great data. Do you know if there is a role for B cells in this integrated immunity? Have you tried experiment where you have depleted B cells, and are the mice still?
We did do the mu-MT mice where we depleted the B cells. And in this model, we didn't see an impact, but I think, I think that needs to be explored very well. I mean, Taya, has this beautiful data on antibodies activating or suppressing the innate system through FC receptors. I'm sure they play into it as well. Yeah.
And we have time for questions. Oh, okay.
Thank you. As you know, there's data from not only BCG, but from other vaccines on decreasing coronary artery disease. And I'm wondering, have you looked at the effect of plaque formation in your animal studies? We've looked at that in HPV vaccination, and there is a strong signal that it may prevent deaths by preventing inflammation.
Yeah, it's a good question. We haven't looked at this in this model. However, if you remember, I showed you that vaccines are inducing epigenetic imprints in bone marrow myeloid cells and in monocytes. Of course, monocytes go everywhere. They go to the heart, they go to the brain, they go to the intestine. So it's a plausible hypothesis that vaccines might modulate the epigenome of monocytes to tweak inflammation. And these monocytes, when they seed various tissues, may either have a detrimental or beneficial effects on inflammation as it applies to the heart and the brain elsewhere as well. We're looking at that. So, yeah. 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 Yeah.
I have a much easier time imagining a cell state that's sort of protective against a wide range of different viruses, but why would this protect against allergy?
Yeah, it's a great question. The effect on allergy is the fact that this is such a Th1 polarized environment. And we think that that's how it's skewing the response away from a Th2 to a Th1. So I showed you reduction in IL-4, IL-5, IL-13 in eosinophils and IgE. If you looked at the allergen-specific Th1 response, there was initially a little bit to begin with. If anything, it seems to have increased. So I think a working hypothesis, and we have to dive into the precise mechanism, but how we think is protecting against allergy is basically skewing the Th2, Th1 balance.
Could you square your data with a hygiene hypothesis? Basically, 100 years ago, what you induce with your universal vaccine right now was done by more infections that we underwent?
Could be, I mean, it's entirely possible. I mean, it just goes to show that prior imprinting exposure, either through pathogens or whatever, really has an effect that we don't really understand mechanistically. And I think it needs to be looked at in the context of modern day vaccines. So,
Thank you, Bali.
thank you.